Hello, today I will be talking about all of the topics that were covered in the first half of cell and molecular biology. So, what I will be covering is chromatin compaction in the roles of HDACs and HATs in DNA accessibility, DNA replication, DNA mismatch repair and the two mechanisms used to repair DSPs, the purpose of the pre-initiation complex, also known as PIC, the post-transcriptional mRNA processing, translation, all of it, combinatorial control in the mediator, and RNAi. In this section, I'll be talking about chromosomal DNA and how it's packaged. I'd like to have a disclaimer that the picture is blurry, but I thought it would be a great picture to help explain the topic. So, there are about 3.2 billion base pairs in the human genome, so one chromosome can contain millions upon millions of base pairs in them. For example, if you were to stretch out the entire human genome, you would be able to go to the sun and back about 300 times. That's crazy. So thinking about that, how are we able to package all of that into our cell? DNA is linear and would be extremely long, and that would never fit into such a small nucleus. Imagine trying to put 24 miles of fine string into a tennis ball. Almost impossible, huh? Well, the DNA needs to be packaged before it is put into the nucleus, and that is done by many different specialized proteins that bind to the DNA and cause it to fold up, which allows for the DNA to become compact. In this picture, the white string is the DNA, and it's being wrapped up by those yellow cylinders, which are also called histones. Histones are small proteins that are used to help pack up the DNA. A nucleosome is a stretch of DNA coiled around histones. So in the picture, each yellow cylinder wrapped up with the white string is a nucleosome. The DNA strand is constantly wrapped around those histones, which cause the histones to bunch up, almost like when you wrap a beaded necklace around your finger and make it coil up. The DNA and specialized proteins together are called chromatin, which are nucleosomes coiled and stacked together. So how exactly is the DNA coiled together? Didn't I say earlier that DNA was linear? Yes, DNA is linear, but there are some specialized proteins that do cause it to coil. The H1 or histone 1 is one of the 5 histone proteins that are components of chromatin. H1 is not part of the nucleosome like the other 4. Instead, it sits on the linker DNA, which is the DNA between each nucleosome, right near the nucleosome, and it allows for the zigzagged 30 nanometer fiber to keep its shape. Without the help of H1, the DNA would not be able to coil up, which will not make the 30 nanometer fiber, so the DNA cannot fit into the nucleus, which means that the cell will die. If the 30 nanometer fiber is not made, it can be quite fatal. A lot of histones wrap into the 30 nanometer fiber, which consists of nucleosome arrays in their most compact form, also known as heterochromatin. In the picture, you probably noticed that it says histone octamer, and you're probably wondering, what is that? A histone octamer is the 8 protein complex found in the center of the nucleosome, and it consists of 2 copies of each of the 4 histone proteins, while the 4 remaining, which are H2A, H2B, H3, and H4. The octamer only comes together when a tetramer, which has 2 copies of H3 and H4, comes together with 2 H2A and H2B dimers. So going back to the picture I previously used, you probably noticed HAT and HDAC in a blue and pink oval. A HAT, also known as histone acetyltransferase, is an enzyme that will acetylate conserved lysine amino acids on the histone proteins. What it does is it transfers an acetyl group from acetyl-CoA to create acetylysine. As you probably remember, DNA is wrapped around the histones, and by bringing an acetyl group to the histones, genes can be turned on and off. So, in general, histone acetylation will increase gene expression. So, when talking about HDAC, or histone deactylase, it's a class of enzyme that actually removes the acetyl group from acetylysine, which is on a histone, and allows the histones to wrap up more tightly, which actually is the opposite of what a HAT does. So, when talking about HATs and HDACs, it's a never-ending cycle of the HAT opening up the DNA and adding the acetyl group in order for the genes to be expressed, and the HDACs will close up the DNA and remove the acetyl group so the DNA can once again become tightly wound. So, in this slide is a great overview of everything that I just went over, including the creation of the nucleosome with the histone and the DNA at step 3, and it also shows where the H1 histone will actually land on the linker DNA right next to the nucleosome in order to cause it to coil up into the 30 nanometer fiber, which is shown at step 5, and from there, the DNA will become compressed and start to fold, and then that's where it turns into a chromosome. Now I'm going to be talking about DNA replication. Here's a picture of a replication bubble, which is an unwound and open region of DNA where the replication occurs. The bubbles are created when helicase, which is an enzyme, separates the two strands of DNA so that they can be replicated. You can also see the replication fork, which I will zoom in on. So here's the picture of the replication fork. I know it looks like a lot, but I'm going to break down every single part. So before DNA can be replicated, it has to be remembered that it needs to be unzipped into two single strands. DNA has 4 bases called thymine, adenine, guanine, and cytosine, and there are pairs between the two strands. Adenine will only pair with thymine, and guanine will only pair with cytosine. And in order to unwind the DNA, the interactions between the A and T and C and G's need to be broken. This is performed by DNA helicase, which will actually interrupt the hydrogen bonding between the base pairs. Once the hydrogen bonding is then separated, the strands will turn into a Y shape known as the replication fork. This is where replication will begin. It's important to remember that DNA is directional, and it's signified by a 5' and a 3' end. The 5' end has a phosphate group, while the 3' end has a hydroxyl group. It's important because replication only progresses in the 5' to 3' direction. However, it's important to remember that the replication fork is bidirectional. So, looking in the picture above, the top strand going from 3' to 5' is the leading strand, and the bottom strand going from 5' to 3' is the lagging strand. I'll explain why in a little bit. So, when talking about the leading strand, which is seen at the top of the picture, it's actually the easiest to replicate. A primer, which is a piece of RNA, will bind to the 3' end of the strand, and it will always bind as the starting point for replication. And the primers are generated by RNA primase. The lagging strand actually begins replication with multiple primers, and each primer is only like a couple or a good amount of bases apart from each other. The DNA polymerase, which I will talk about in a second, will add pieces of DNA, which are called Okazaki fragments, and you can see the 3 Okazaki fragments on the lagging strand, and the Okazaki fragments are between the primers. This way of replication is actually discontinuous because the new fragments are disjointed and they need to be brought together using DNA ligase. You can actually see the DNA ligase, which is like a purple diamond, joining all of the fragments together to create one unified strand. Going back to DNA polymerase, it's actually responsible for creating new strands of DNA by a process called elongation. I'll be focusing on polymerase 3, which is the main replication enzyme, and the other polymerases 1 through 5, excluding 3, are actually responsible for error checking and repair. I'll cover that in one of the next couple of slides. So what DNA polymerase 3 actually does is that it binds to the site of the primer on the strand, and it will begin adding new base pairs that are complementary to the original strand during replication. So going back to the leading strand, because replication goes in the 5' to the 3' direction on the leading strand, that's why the strand is continuous. Once the continuous and the discontinuous strands are complete, DNA polymerase 1 will then get rid of the RNA primers and then replace them with the appropriate bases. It pretty much proofreads the newly formed DNA to check and see if it has to remove or replace any errors. Once the DNA polymerase 1 is done, that's where the DNA ligase will join the Okazaki fragments to form one single strand. Once DNA ligase has done its job, there's still a problem. Because the DNA polymerase can only add nucleotides in the 5' to 3' direction, the ends are out in the open. So at the ends of the parent strands, there's a sequence called a telomere. Telomeres act as protective caps at the end of the chromosomes to prevent other chromosomes from fusing together. Oh my gosh, I almost forgot one of the most important parts. The sliding clamp. What the sliding clamp does is that it's a protein that binds to the DNA polymerase and prevents it from disassociating with the template DNA strand. When talking about the clamp loader, it requires ATP to close the clamp around the DNA. The clamp loader will also load DNA into the ring-shaped clamp in order to get the polymerase to the DNA. So now I'm going to be talking about DNA mismatch repair. What DNA mismatch repair is that it's a system for recognizing and repairing wrongful insertion, deletions, and misincorporations of bases that can happen during DNA replication and even DNA recombination. It's also good to remember that mismatch repair is actually strand specific. So looking in the top left corner of the picture, you can see that there is a mismatch and the incorrect base is on the daughter strand, while the parent strand has the correct base. The reason for this is because the parent strand is used as a template, and it knows that it's correct because the parent strand has already been methylated, which is like a sign to show that it made it through inspection. So how does mismatch repair work? First, a group of proteins will recognize and bind to the mispaired base. A second complex will then cut the DNA out around the mismatch, and then more enzymes will come in and chop out the incorrect nucleotide. A polymerase will then replace the missing section with the correct nucleotide or nucleotides, and DNA ligase will seal the gap. I mean, it seems pretty simple, but it's much more complicated than that. There is a repair process called base excision repair, which is shown here, and there are 5 steps: the excision of the base, the incision, the end processing, and repair synthesis, which includes the ligase filling the gaps. So that picture was a base excision repair, which actually just removes the damaged base, while the picture that I showed first was nucleotide excision repair, which is where parts of the nucleotide are removed. So there are 2 mechanisms in order to fix DSBs, also known as double-stranded breaks. In the picture on the left, the process used is non-homologous end joining, and on the right is homologous recombination. So I'm going to be focusing on non-homologous end joining first because the process itself is quite short and pretty simple. So in the recognition stage, you can see that there are 2 breaks on the end of both sides of the DNA. So what happens is that those ends are shaved down, and then the DNA is ligated together. The reason it's called non-homologous is because the break ends are actually directly ligated without a need for a template or a homologous template. Non-homologous end joining is actually quite violent, but DSBs are actually one of the most dangerous forms of DNA damage, so it needs to be done. So now if we focus on homologous recombination, it's a type of recombination with which the nucleotide sequences are actually exchanged between 2 identical molecules of DNA or even 2 similar molecules of DNA. So if we focus on the picture, at the top you can see the DSB, or the double-stranded break. Then what happens is that the 5' ends are shaved back to create 3' overhangs. Once the 3' overhangs are created, they will then begin to invade, in a process known as strand invasion, the other molecule. When the 3' ends actually invade the other cell, they create DNA crossover, or a holiday junction. And they will also provide a primer to start the new DNA synthesis. Let's focus on the blue 3' end that it invades. You can see that once it gets down to the molecule, you can see that synthesis begins, which means that there was a primer at the end that allowed it to start synthesis. I almost forgot. When the blue strand invades, it causes the orange strand to bubble up because the blue wants to interact with the base orange line, and it can't do that once the other orange line is actually there. So once DNA synthesis is complete, you can see these little blue arrows right near the crossing over at the junctions, or the holiday junction. So what the blue arrows actually stand for is it means that the DNA is actually cleaved right in the middle of the X. So the upper half of the X will go upward and the bottom half will snap downward. The reason for this is because DNA is actually linear and it cannot stay bent for too long. DNA actually hates being bent, so anytime it has the chance to become linear again, it will take it. So once the DNA actually snaps up into place, you can see with the dashed line where the newly synthesized DNA, which was actually synthesized by DNA polymerase, is. And with the green arrows, that tells you exactly where the DNA is ligated, because there are still spaces in between the new and old strands, and if there is a space in the DNA, it could be quite fatal. So now I'm going to be talking about the pre-initiation complex, and once again, I know the picture is quite blurry, but it's a great picture to help describe what exactly goes on. So what is the purpose of the pre-initiation complex? Well, it's necessary for transcription, and what it does is it positions RNA polymerase at gene transcription sites or start sites, and then it will denature the DNA, and then it will position the DNA in the RNA polymerase 2 active site, and then that will begin transcription. So, what is the purpose of the pre-initiation complex? Well, you can see the core promoter, and in the promoter you can see the TATA box, which is actually very important. And before I start with all the parts, I want you to remember DAB-Phe. That is the order of how everything is added. So what happens first is that the TATA binding protein, also known as TBP, which is found in TFIID, will then bind to the promoter, and it'll cause a bend in the DNA. Next, the TATA binding protein will then interact with TFIIA, and then it'll cause TFIIA to come to the promoter. The TATA binding protein will also interact with TFIIB, which will then allow TFIIB to be recruited to the promoter, and that is DAB and part of DAB-Phe. Those three have already been recruited. And then there will be interactions between TFIIB and TFIIF and the RNA polymerase 2, and the interaction between TFIIB and TFIIF will recruit the RNA polymerase to the promoter and TFIIF. I like to think of TFIIF as like a little hat on the DNA polymerase because they're recruited together. So that I like to say that's why there's a gap in DAB-Phe. TFIIE will then join the complex and will also recruit TFIIH, which is the final one, and it has a protein kinase activity which will interact with the RNA polymerase 2 and DNA helicase activity. There are certain areas of TFIIH that will have ATP and helicase activity, and it causes a negative tension in the DNA. And what that DNA tension causes is the DNA to unwind, and it'll form the transcription bubble, which I talked about earlier. So the template strand will engage with the RNA polymerase to an active site, and I like to think of it as the RNA polymerase almost shoots out and begins RNA synthesis. So now I'm going to be talking about the post-transcriptional mRNA processing. So transcription occurs in the nucleus of a cell, and the RNA that's synthesized in the process is then transferred to the cytoplasm, where then it is translated into a protein. However, it's not that easy. The RNA from transcription is not immediately ready for translation. So the modifications of the RNA after transcription have two purposes. The modifications will help the RNA be recognized, and then it will be translated into proteins. And during the processing of the transcription RNA, there are portions of the RNA that are not supposed to be translated, and then they will be cut out. By cutting out those unwanted sequences, it allows protein synthesis to be quite efficient because it'll focus on one specific protein to code, and it won't have to focus on all the unwanted ones. Then, if it was to focus on the unwanted ones, the synthesis would be slowed down significantly, and it would take a lot longer for translation to recognize RNA. Now that I talked about why everything happened, I'm going to be focusing on the modifications, which are the 5' capping, the addition of the poly A tail, and the splicing. So looking at the picture above, the red lines are the exons and the blue lines are the introns. The exons are pretty much all that is needed for translation, so there has to be a way to get rid of the introns. So what happens is the introns will then bubble up into like a little bubble looking thing, and they will be excised out of the mRNA, and then the exons will be ligated together. So this picture is actually a really good picture because you can see an overview of almost everything that happens. There are some topics that I'll talk about later in the video, but inside of the nucleus you can see where transcription occurs in the creation of pre-mRNA and the introns and the extrons, and then the exclusion of the introns, and you can see the little word cap and a whole line of A's. So the cap replaces the triphosphate group at the 5' end of the RNA and with a special nucleotide which is actually known as the 5' cap. The 5' cap is believed to help the mRNA recognition by the ribosome during translation. There's also another modification that takes place at the other end of the RNA, known as the 3' end, and to the 3' end there are about 30 to maybe even 500 adenines added to what's called the poly A tail. So once the introns are removed and the cap and the poly A tail are added, the mRNA is ready to go into the cytoplasm. Once in the cytoplasm, the cap will receive a cap binding protein and the poly A tail will receive a poly A binding protein. So the purpose of the poly A binding protein is actually to protect the tail from degradation, and it can also regulate mRNA production. Without the poly A binding protein, the poly A tail would not be added, and the RNA would degrade at a rapid rate. So the cap binding protein is actually very similar to the poly A binding protein, except it's found on the cap. So when in the cytoplasm, if everything went right, like all the introns were removed and the cap and the poly A binding protein were correct or in the correct places, the DNA will circularize and become a circle, like shown at the top of the picture. So by becoming circularized, that means that the mRNA is ready for translation. So now I'm going to be talking about translation. Well, all of the parts of translation. So what is translation? Well, translation is the process of creating proteins from mRNA, which acts as a template. So on the RNA, there are nucleotides, sequences of nucleotides that are translated into amino acid sequences of proteins, and those reactions are carried out by the ribosomes, which are the green structures in the picture. So ribosomes in tRNA, which are the purple and blue looking stamp things, they dock onto mRNA transcript, and they will recruit multiple enzymes in a process that uses a whole lot of ATP. So here's a picture of tRNA. So what tRNA does is that it helps decode messenger RNA sequences into a protein. So at the bottom of the picture, you can see the word codon in the sequence GCC. It is GCC because remember, DNA is read from 5' to 3'. So above it, you can see anticodon, which is actually on the tRNA, and you would think it would be read CGG, but it is actually read GGC because remember, it is read from 5' to 3', and the anticodon is the opposite of the codon. So the codon and the anticodon have to match. So if the tRNA sees GCC, then it knows that it needs an anticodon of GGC in order to match it and create an amino acid. That certain sequence gives us a specific amino acid, and I will go into what exactly is used in order to see what amino acid is made. So here's a picture of the genetic code, and this helps us figure out what amino acid is being created. Since stretches of 3 nucleotides can create a total of 64 codons, one single amino acid can be coded by multiple codons. You'll notice in the picture above that it's very rare to have one codon just code for one single amino acid. Most of the time it's either 2, 3, or 4, but it is very rare to see just one. Here is a close-up of a ribosome, and the ribosomes are pretty much the primary machines that drive protein synthesis. A ribosome is made up of 2 subunits, and translation starts when the smaller subunit will bind to the sequences that are upstream of the coding sequence on the mRNA. The smaller subunit will then recruit the large subunit of the ribosome, and then translation will begin. So the ribosome contains 3 very important regions, the P site, the A site, and the E site, and they are formed by the 3-dimensional shape of our RNA. The P site will bind to the growing polypeptide, and the A site acts as an anchor for incoming charged tRNA after the peptide bond is formed. The tRNA will bind briefly to the E site before leaving the ribosome. So translation will begin when the cap on the mRNA is recognized by the smaller subunit of a ribosome. That ribosomal subunit, with tRNA, will then start to scan the mRNA for the start site of translation, which is often AUG, which codes for methionine. The ribosome and the initiator tRNA will then slowly move along the mRNA until they can find the start codon. What the tRNA does is that it ensures that it's recognized by the initiation factors, but it wants to be discriminated against by the elongation factors. So once the larger subunit of the ribosome arrives, the tRNA will go to the P site, and that's really where translation starts. When the initiator tRNA is binded to the P site, the ribosome will bring it close to the amino acid or the A site of the ribosome, where the next codon will actually wait for translation. New tRNAs carrying amino acids will enter the ribosome at the A site, and then go to the P site, and then go to the E site. The reason for this is because, remember, it's always read from 5' to 3', and the anticodon is anti-parallel of the mRNA, so it's actually going from right to left, 5' to 3'. By using complementary base pairing, the correct tRNA will bind to the ribosome. The ribosomal RNA actually catalyzes a peptide bond between the amino acids in the A site and the P site. And for example, if you were to take the purple dot and the orange dot above, you put a peptide bond between the both of them and connecting them. And then the bond between the P site and the orange dot will then be destroyed. Once the orange dot and the green tRNA are not connected anymore, that tRNA will then leave, and the blue tRNA in the A site will then move over to the P site, which will leave the A site open for another tRNA. So the process of connecting the amino acids in the P and A site, and then the tRNA leaving, and the one in the A site moving to the P site, will continuously happen until a stop codon is reached. So when a stop codon is found in the A site, it's recognized by release factors, and they will then attach a molecule of water to the polypeptide chain rather than another amino acid. This causes the process of translation to stop, and it will then release the polypeptide chain from the ribosome. Both subunits of the ribosome will then dissociate and get ready for the next cycle of translation. I actually forgot to add that the tRNA cannot actually just randomly move to the E or the P site on its own. What happens is that the bottom of the ribosome will start to move, and then the top of the ribosome will almost click over to fit up with the smaller unit again, so the tRNA will end up in the E site and then leave if it is empty, and then what used to be in the A site will end up in the P site. I will now be talking about the combinatorial control of gene expression and the mediator. So in the picture above, you can see a lot of what I talked about earlier in this presentation. You can see the promoter, the TATA box, the general transcription factors like TFIID and like DABFA. But there is a new section to this, something called the mediator. So the mediator is a complex that acts as a co-activator. The main function of it is to transmit signals from the transcription factors to the polymerase, as seen as the blue or light blue dot at the bottom of the picture. The mediator will also come in contact with the transcription regulators, as seen as in the top. And what happens is when something comes in contact with the mediator, it'll change shape and start to interact with other molecules based on which shape it's in. You can almost say that the mediator is like a bridge. It will listen to everything that the transcription regulators have to say in like their regulatory DNA sequences, and then it will pass the message on to the transcription factors in the polymerase. You probably noticed how there is DNA just pushed off to the side and just wrapped up, and that's because it allows for the transcription factors to come in contact with the mediator. Without that, they would have a really hard time trying to get in contact with the mediator. Going back to combinatorial control, I didn't really describe what that is. What it is that any given gene is most likely controlled by a specific combinations of factor that control transcription. The factors actually control gene expression from various DNA enhancers in a combinatorial fashion. So enhancers and silencers are a very good example of that. Both silencers and enhancers can control regions of DNA that could even be thousands of base pairs away from them or the gene they control. A silencer will actually repress the expression of the gene they control. However, the enhancers will do quite the opposite, and they will continuously express the gene they control. So this is a great picture to explain what an enhancer and a silencer are. So the enhancer will be activated if there are low levels of something needed in the DNA, so then it will be turned on and the gene will be expressed at a high rate. The silencer will turn on and it acts as a repressor. So if the levels are super high and no more needs to be made, then the silencer will be turned on and the gene expression will stop. So going full circle, why would we need to keep the gene on if there are already high levels of something? We don't, and that's why the silencer is there. And we need to make something if there are low levels, and that is why the enhancer is there. Going back to the mediator, this is what actually happens when the transcription regulators come in contact with it. What they do is they see if there are levels if the levels are high or if they're low, and if the enhancer or the silencer need to be turned on. They'll touch the mediator and cause it to change shape, which will then tell the mediator to tell what's needed to turn on and off. So I'm going to be talking about the final topic, which is RNAi. RNAi is a mechanism used by cells for gene silencing, and it requires the formation of double-stranded RNA. Once the double-stranded RNA is recognized by the RNAi machinery, all of the ssRNA and the double-stranded RNA will be destroyed. It is important to keep in mind that all of the ssRNAs and the double-stranded RNAs with the same sequence are actually destroyed. So, if there's a double-stranded molecule with a sequence that's in my gene and it's recognized by RNAi, then I'm never making a protein from that gene again. So, if any mRNA is transcribed from the gene, it will be immediately marked for destruction or it will be immediately killed. RNAi is a very potent response, and it's believed that it was used against viral infection because viruses are primarily the natural source of double-stranded RNA. So, RNAi is actually triggered by the double-stranded RNA that is fully base paired and is about 23 base pairs long. If it is longer than 23 base pairs, then Dicer will come in and cut them up into small siRNA, which are small interfering RNAs. The siRNAs are then bound to RISC, which is the silencing complex in RNAi. And RISC will use one of the two siRNA strands and bound to it to recognize other RNA molecules, either ssRNA or double-stranded RNA, with the complementary sequence. Once RISC actually silences the gene and pretty much gets rid of it, anytime it sees the complementary sequence, whether it be in another viral RNA or if it's in anything pretty much, it will destroy them immediately. Pretty easy, huh? RNAi is honestly one of my favorite sections of this class because it immediately clicked in my head right after learning about it in class. Well, this marks the end of my first ever eReport. I hope you enjoyed, and thank you so much for watching.